Hello everyone, I am Dr. S. Rajesh, Associate Professor in Biotechnology from Center for Plant Molecular Biology and Biotechnology from Tamil Nadu Agricultural University, Coimbatore. And today we'll be discussing on the genetic engineering technology, also known as recombinant DNA technology for expression of foreign genes in biological host systems. To do so, we particularly need a type of enzymes called as DNA manipulative enzymes. And in this particular lecture, we'll be seeing in detail about different DNA manipulative enzymes available for construction of recombinant DNA and how they are used for DNA manipulation. So genetic engineering basically is an enabling technology which is used for cutting, modifying, and joining DNA by using specific enzymes like restriction endonucleases and ligases that will be used to generate DNA fragments as well as join them in a vector system which act as a vehicle to carry the foreign gene of interest. Once the recombinant DNA is constructed by recombining the genes from two different organisms, preferably from two different organisms, they can be mobilized into or introduced into a host cells for expression of the recombinant proteins, expressing the particular target gene which we have mobilized into the host system. This particular technology, genetic engineering or recombinant DNA technology, has plenty of applications in the basic research as well as in medical sciences, in industrial applications, as well as in agricultural sectors. However, despite the umpteen number of advantages we have for this technology, this technology also raises some concerns with relevant to ethical, legal, and safety issues. First, let's see what is genetic engineering. Genetic engineering is a process that uses laboratory-based technologies to alter the DNA makeup of an organisms. That's why we also call this technique as genetic modification. Now talking about a recombinant DNA technology, the recombinant DNA in particular, the DNA molecules which are constructed outside the living cells by in vitro, means in the test tubes by joining either the naturally occurring DNA sequences, or we can also go for joining together the synthetic DNA fragments which can be replicated in a living cells to produce the recombinant proteins. The objectives or goals of this recombinant technology is to isolate and characterize a gene and to make desirable alterations in one or more isolated genes by using the DNA manipulative enzymes, more particularly the enzymes like restriction endonucleases as well as DNA ligases. The third objective is to return back the modified or altered genes back into the host or living cells to propagate the cells containing the recombinant DNA to express the foreign protein or recombinant proteins coding for the target genes which we have mobilized. This is also called as gene cloning methods. So talking about DNA manipulation, this is a basis of recombinant DNA technology, which relies in manipulating the DNA using specific enzymes under in vitro conditions. All the DNA manipulative enzymes normally use a purified form of enzymes. Within these cells, these enzymes, those naturally occurring enzymes involved in DNA replication as well as transcription and repair of the DNA molecules, and some of the enzymes were also used in regulatory mechanisms. Talking about the types of DNA manipulative enzymes, broadly, these manipulative enzymes can be classified into four different types, like nucleases, ligases, polymerases, and the DNA modifying enzymes. And we will be discussing about each and every different types of DNA manipulative enzymes. The first one is nucleases. As the name implies, it is an enzyme that will act upon the nucleic acids. These nucleases degrade DNA molecules by breaking the phosphodiester bonds, which links the nucleotide bases one after another in the DNA strand. And these nucleases are specific for the DNA as well as RNA. The template on which it acts upon, the nucleases are accordingly classified. And there are two different kinds of nucleases. One is called as exonuclease, the other one is endonucleases. The exonucleases They remove the nucleotides one at a time from the end of the DNA molecule. The primary difference between the different exonucleases lies in the number of strands that can degrade when a double-stranded molecule is attacked by the nucleases. For example, the BAL31 enzyme, which removes the nucleotides from both the strands of double-stranded DNA. Here you can see the double-stranded DNA is acted upon by the enzyme, which cleaves the DNA on both the strands. It acts upon both the leading and lagging strands of both the strands of DNA. The second enzyme is E. coli exonuclease 3, which removes nucleotide only at the 3' end. In both the cases, both the strands, you can see typically they remove the nucleotide bases only at the 3' end of the double-stranded DNA molecule. The third type of exonuclease is lambda exonuclease. These exonuclease remove DNA at the 5' end. Like exonuclease 3 removes 3' end, the lambda exonucleases remove the bases only from the 5' end of the DNA. The second category of nucleases is endonucleases. So they, as the name implies, endonucleases, the nucleases will act upon the internal regions or the internal phosphodiester bonds of the DNA molecule. For example, the S1 endonuclease enzyme, it cleaves only the single-stranded DNA, which includes the single-stranded nicks that cause mainly in the double-stranded DNA. The second type is DNase 1 or deoxyribonuclease 1, which will cleave both single-stranded as well as double-stranded DNA. When we talk about endonucleases, there are special category of enzymes within this group called as restriction endonucleases, which cleaves the double-stranded DNA by making a nick or cut at specific recognition sequences. And that's the reason we call this enzyme as molecular scissors in the recombinant DNA technology. These particular restriction endonucleases are found in bacteria as well as in archaea, and they provide a kind of defense mechanism for these organisms when they are challenged by the virus infections. Restriction endonuclease, the major kind of nucleases or the DNA manipulative enzymes, was first observed in the laboratories of Werner Arber and Matthew Michelson in 1960s. When they were working in the laboratory, they found that the restriction is caused by the enzymatic cleavage of the phage DNA. Those are all bacteria-eating viruses, and the enzyme involved was therefore accordingly termed as restriction enzyme at a later date. The term restriction endonuclease was coined by Liederberg and Michelson in 1964. And if you see the historical milestone subsequent to the discovery of the enzyme by Arber and Michelson, the Smith and Nathans, they have discovered this restriction endonuclease from Haemophilus influenzae. And more particularly, there are two enzymes that were discovered by these people. One is HIND1 and HIND2. So these restriction endonucleases have a specific or a typical nomenclature for naming them. Normally, these enzymes start with the genus name, the first letter of the genus name, followed by the two letter of the species name. Say, for example, in case of Hindi3, it denotes H, the genus, Haemophilus, the two letters of the species influenzae, IN, D1, that denotes the order of discovery of a particular strain from which the enzyme is derived from. So we have many number of restriction endonucleases. For example, we have more than 2,500 restriction endonucleases that are being isolated and characterized. Some of the examples include the Serratia marcescens, one enzyme, and typically they have a recognition sequences. And we have Haemophilus aegypticus, Haemophilus influenzae, Hindi3, Hindi2, Hindi3, and BAMH1 from Bacillus amyloliquefaciens. So as I mentioned, these enzymes are very specific and they cleave the DNA at an appropriate recognition sequences. So these restriction endonucleases has typical characteristics where depending upon the enzyme they cut, they are being classified into 4 different types: type 1, type 2, type 3, and type 4 enzymes. Among these types, the type 2 enzyme is widely used in the genetic engineering applications. Now let's see the different types here. The type 1 enzyme cleaves the DNA at random sites, more particularly from far away from the recognition sequences, and they have a limited application in that case. The second type which is widely used in the genetic engineering. It cleaves the DNA at a very defined positions, very close to or within the recognition sequence of the template DNA. So within the type 2 restriction endonucleases, we have three subclassifications, that is 2G, 2P, and 2S. So these type 2G restriction enzymes, they cleave outside the recognition sequence with both restriction endonuclease as well as the methyl transferase enzyme activities that can act upon in the same protein. The second enzyme within the type 2 is type 2P, which cleaves the symmetric targets, whereas the type 2S enzyme, they recognize the asymmetric sequences of the template and attack them. The third type, the type 3 enzyme, restriction endonuclease enzyme, it cleaves outside its recognition sequence, and they require two sequences in opposite direction within the same DNA. The fourth type, that is the type 4 restriction endonuclease, they cleave the modified or methylated DNA sequences. So I repeat, we have four different types of restriction endonucleases. Among the four different types, the type 2 restriction endonuclease is the most widely applied used enzyme in the genetic engineering applications. So here I'm just displaying some of the restriction endonuclease and their recognition sites. So these restriction endonucleases are classified as enzymes depending upon the number of bases they identify or recognize and cut. And some recognize 4 bases, we call them as 4-base cutters. And some recognize 6 bases, we call that hexameric, they 6-base cutters. And some are 8-base cutters. So depending upon the number of bases they recognize, we call them as base cutters, 4-base, 6-base, or 8-base cutters. So this picture shows how the DNA fragments can be generated that can be used for cloning into a vector or vehicle system. For example, when you use restriction endonuclease for cutting the template DNA, the enzyme can cut the template DNA and can generate fragments which may have a blunt end, or sometimes they may act upon the template DNA at different position within the restriction recognition sequences, and they may generate protruding ends. In this case, this is producing a 3' protruding end, and in case of EcoR1, it generates a 5' or 5' protruding ends. So when we compare this blunt end and protruding ends, the blunt end is also called as non-sticky ends, and protruding ends are sticky ended fragments. And these protruding ends are very much amenable for generating recombinant DNA in the cloning process. There are a few considerations when we choose the enzymes to manipulate or cut or restrict the DNA fragments. And say, for example, let's assume a DNA molecule with 50% GC content and with a random distribution of the 4 molecular alphabets. A 4-base cutter will be able to cut the DNA molecule into fragments once in every 256 nucleotide pairs. That is 4 power 4. That denotes the 4 molecular alphabets ATGC. And the 6-base cutter will recognize every base to cut at 4096 nucleotide pairs, and similarly a 8-base cutter will recognize that's 4 power 8, that's 65,536 nucleotide pairs. These restriction enzymes, I told you the type 2 more particularly is very widely used in genetic engineering, and these enzymes have recognition sequences which may be very similar for some of the enzymes. Some restriction enzyme may have or identify a similar recognition sequence. In such case, those restriction enzyme from different sources but having a same recognition sequence are called as isoschizomers. For example, the Moraxella boris MBO1 and the Staphylococcus aureus SAU3A enzyme, both the enzymes from 2 different bacterial sources, they recognize the sequence GATC. There is another terminology called as neoschizomers, where it is like isoschizomer with the same recognition sequence, but the place where they make cut or restriction varies within the same recognition sequence. We call them as neoschizomers. The example being AAT2 and ZRA1 acting as neoschizomers. The second DNA manipulative enzymes widely used in the DNA manipulation is ligases. And these ligases, typically called as molecular glue or paste, which are used to join the DNA fragments of various types we generate. And this is one among the fundamental steps in the recombinant DNA technology. And this process of fusing together the DNA fragment is called as ligation. And this is achieved by the catalytic reaction of the enzyme ligase. Normally, the enzyme ligase act upon the fragments or template DNA to join them by spending at the expense of an ATP molecule. They are energy spending reaction that catalyze the bonding between the phosphodiester bonding between the two fragments. For example, the E. coli DNA ligase and the Phage T4 DNA ligase are two major sources which are being commercially available for use by the genetic engineers. Here you can typically see the ligase activity. When we generate the insert DNA by specifically using the gene-specific primer to act upon the template DNA from any host DNA, maybe a genomic DNA, you are pulling out a particular fragment of the gene by using gene-specific primer in a polymerase chain reaction. This insert DNA has to be modified with some enzymes, this DNA manipulative enzymes, so that they are amenable for cloning in a vector system. In this case, when this PCR or polymerase chain reaction is executed by using a common enzyme source polymerase from Thermus aquaticus, this TAC DNA polymerase typically generates the overhangs which can be used to clone in the TA vector systems. For example, when we amplify the DNA fragment in a PCR system using the TAG DNA polymerase, at every cycle of reaction, the extension step, they generate the fragments with A tailing, addition of adenine residues in the 3' N. And when you choose a vector, a TA vector with a 5' N, and the T and A can pair together and perfectly make a ligation reaction possible. And they can be fused together. The insert generated from the PCR reaction can be used to fuse with the opened vector, which is cut with a restriction enzyme, and they can be combined by using the molecular glue called as ligase. As I told you, this ligase activity needs energy spending reaction. By using ATP, they recombine to form a recombinant DNA. The third DNA manipulative enzyme is polymerases. And we have different types of polymerases. There are 4 classes being used in that. The first one is DNA dependent DNA polymerase. The second is RNA dependent DNA polymerase. The third is DNA dependent RNA polymerase, and the template independent polymerase enzymes. So these 4 are different DNA polymerase enzymes used in the DNA manipulation process. Let's see one by one. The first one is DNA dependent DNA polymerase enzyme. This is also called as a Kornberg enzyme or holo enzyme with a molecular weight of 111 kilodaltons. And these enzymes typically upon excision or catalytic action, they generate a larger and a smaller fragment. We call them as a clino fragment with 766 kilodalton, which possess both polymerase activity as well as the exonuclease activity. So this picture will typically show you when the DNA polymerase enzyme 1, it is subjected, it's a holo enzyme. When it is subjected to proteolysis, this will generate 2 fragments. One is a large fragment, the other one is a small fragment. The large fragment contains 2 activities. That is the 3' to 5' exonuclease activity as well as polymerase domain. You can typically see the polymerase domain and the exonuclease domain. The second small fragment is 5' 3' exonuclease domain. So here this image shows the action of the DNA polymerase enzyme at the recognition sequence for the subtlesin cleavage site, which generates 2 different fragments of a smaller with 35 kilodalton, the other one with the 76 kilodalton size. The larger fragment is what I told as clino fragment. So these enzymes, particularly the exonuclease activity and the polymerase activity of the DNA polymerase, can be exploited for polishing the overhanging ends. Typically, you can see when you have an overhanging end, these ends can be filled or polished by using the polymerase activity of the DNA polymerase 1. So this exonuclease activity of DNA polymerase 1, that is 3' to 5' activity, is typically a proofreading activity where you can convert a blunt-ended DNA or you can make some repair arrangements after mechanical fragmentation using the enzymes. And similarly, the 5' to 3' exonuclease activity can be exploited to make a nick translation, which will replace a part of a pre-existing strand of duplex DNA with the newly synthesized molecules. So this DNA polymerase 1 possesses both the proofreading activity as well as the polymerase synthesizing activity. The second type of enzymes within the DNA polymerase is RNA-dependent DNA polymerase, which is also called as reverse transcriptase. And this uses RNA as a template to convert or synthesize DNA. We call them as complementary DNA or copy DNA. And these enzymes are widely isolated from the retroviruses, means the viruses that contain RNA genome. There are two commonly used enzymes sources available. One is from the murine Maloney leukemia virus MMULV. The second one is AMV, that is avian myeloblastosis virus. And these enzymes, they need a primer to polymerase, and this enzyme synthesize DNA fragment with an error rate of 1 in 500 nucleotides. Means the frequency is very, very feeble, low. The third type is DNA-dependent RNA polymerase. The enzymes isolated from the viruses, which include the T3, T7, and SP6 enzymes from the specific phages, and they are very highly specific for their promoters. And that's the reason these polymerase or enzymes can be typically used for expression studies. T3, T7, SP6, even in the sequencing program, we use this DNA-dependent RNA polymerases, and they can be deployed both under in vivo as well as in vitro condition. The fourth class of DNA polymerase enzyme is template independent or sequence independent DNA polymerase. The example being the terminal deoxynucleotide transferase, which is used to add the nucleotide at the 3' end. And the second one is tag DNA polymerase, which will add typically the adenine residues at the end of the PCR reaction, which can be deployed for the specific TA cloning of the genes. Among the major classification of DNA manipulative enzymes, the first we have seen is the nucleases, within which we have discussed on the endonuclease as well as exonuclease. The second category we discussed was ligases. The third was DNA polymerases. And the fourth one is DNA modifying enzymes. These are enzymes which are specifically used to add or remove the chemical groups, also called as N modification enzymes. Some of the major DNA modifying enzymes include the alkaline phosphatase, T4 polynucleotide kinase, terminal deoxynucleotide transferase, as well as topoisomerase. Among these, the first three, alkaline phosphatase, T4 polynucleotide kinase, and terminal deoxynucleotide transferase, are widely used in DNA modification under in vitro conditions. The fourth one, topoisomerase, which is been used to relax the supercoils that normally happens during the course of DNA replication or any change that occurs to the double-stranded DNA. So that relaxes the tension created due to supercoiling. They are topoisomerases. So the first in the DNA modifying enzyme category is alkaline phosphatase. This alkaline phosphatase, we have a major sources of isolation of this enzyme called as alkaline phosphatase. It can be extracted, isolated from Escherichia coli, calf intestine, or even Arctic shrimp. And this enzyme act upon the template DNA and removes the phosphate group. Here you can see the template DNA is acted upon by the alkaline phosphatase, which remove the phosphate group from the 5' terminus. When the phosphate group is removed from the pi terminus, they typically at all the 4 base ends, they will have hydroxyl groups only. So here you can see the calf intestine alkaline phosphatase enzyme acting upon the template, where once the phosphate group or moiety is removed, that will prevent the DNA from self-ligation. So normally when the DNA fragments are linearized, are supplied by the firms for cloning purposes, these fragments are going to be subjected to DNA modifying enzymes treatment so that they don't self-ligate. Say, for example, if vectors are supplied by the firms, they will be subjected to DNA modifying enzymes, and the ends will be modified so that they don't undergo self-ligation. So the fragments cannot self-ligate on their own. But once you use a ligase enzyme, The ligase naturally supplies the phosphodiester bonds for fusing the naked DNA, making them as a ligated molecules. So these enzymes are most active at the alkaline pHs. That's the reason we call them as alkaline phosphatase. So there are two primary uses for this alkaline phosphatase enzymes in DNA manipulation. The first one I told you, it's specifically used for removing the 5' phosphate end from the plasmid DNA, which is widely used as a cloning vectors. Even some of the larger DNA fragment can be cloned in the phage-derived vectors. So the first application here is to prevent the self-ligation of the vector. Second, it facilitates the ligation of other DNA fragments into the vector, or we go for subcloning purposes. The second important use for the alkaline phosphatase enzyme is to remove the 5' phosphate from the fragments prior to labeling with the radioactive phosphate. This is particularly we do for random labeling or labeling with the radioactive phosphates for probing purposes. Normally, when we go for blotting techniques like the techniques that are used to prove the presence of DNA or the foreign gene in your integrated into a genome, we go for southern blotting techniques. In that case, if you go for probing your target DNA by using a specific probe, these probe for further detection has to be indirectly for detection, they have to be labeled with the radioactive phosphorus, that is P32 radio labeled probe we generate. And for generating radio labeled probe, first you have to remove the 5' phosphate, and in that place you will be using this radio labeled phosphate, which upon binding them to them, when it identifies the target DNA in the blotting process, they bind to them and emit the radioactivity that helps to trace your presence of gene in the transgenic organisms once they got integrated into. And this alkaline phosphatase enzyme, although they are active in the alkaline pH, they are inactivated by the heat. So here it's a summary of the action of the double stranded DNA, which can be ligated by using the ligase enzyme to create dimers. This normally happens when you do a PCR, we get a PCR dimers when there is a complementary between the two primers we use. Once these enzymes are subjected to treatment with alkaline phosphatase, and the end of the one protruding end of the DNA fragment, when it loses the 5' phosphate, it loses the property of self-ligation. And once you introduce a foreign DNA fragment to construct a recombinant DNA and fuse with the modified vector, these two, when they recombine with the help of the ligase enzyme, the ligase enzyme will supply the phosphodiester bond connection, also the phosphate moiety, which will help in repairing the nicks and create a recombinant DNA. The next DNA modifying enzyme is terminal deoxynucleotide transferase, isolated from the calf thymus source. As the name implies, it is a nucleotide transferase which acts upon the DNA, and particularly in that 3' terminus. Here you can see the nucleotide bases can be added to the 3' end of the DNA by using the terminal deoxynucleotide transferases. So they typically add bases only to the 3' terminus. The third enzyme, DNA modifying enzyme, is polynucleotide kinase. This is isolated from the E. coli bacteria infected with the phages, T4 phages, and this enzyme adds phosphate group to the 5' terminus. So here you can see this is a template DNA where all the 4 ends have a hydroxyl group and there is no phosphate moiety here. And the phosphate can be added, the phosphate group can be added when you treat it with the polynucleotide kinase. So the enzyme activity of polynucleotide kinase is exactly opposite to that of alkaline phosphatase. As if you recollect, the alkaline phosphatase removes the phosphate group from the 5' end, whereas polynucleotide kinase will add phosphate group at the 5' end. The another modifying enzyme is methylases. These methylases DNA basically they protect the DNA from attack by the restriction endonucleases during the cloning process. Normally, when a DNA is being methylated, um, they are prevented from the attack or challenged by the external agents. Maybe it can be viruses, or it can be any other regulating enzymes that will act upon the template DNA to do transcription and express those genes. Say, for example, in case of immunology, the cancer-causing genes in any organism would normally be in a methylated state. That's why we call them as a benign state. Once that benign state DNA is the methylated regions are removed or demethylated, they are now exposed to action by the regulatory enzymes to express them and produce a protein which would finally produce cancer, result in the cancer. These methylase enzymes utilize the substrate S-adenosyl methionine for the methylation reaction. So we have discussed on different DNA manipulative enzymes, the four major classes being the nucleases, which also contains exonuclease as well as endonuclease. The second major enzyme is the ligases, which is used as a molecular glue or molecular paste for joining together of the DNA fragments. The third enzyme is polymerase enzymes. We have two different classes of DNA polymerase enzymes for various applications. The fourth enzyme was the DNA modifying enzymes, which are specifically used in experiments. And although we have different enzymes for manipulating the DNA to generate DNA fragments as well as to cut a vector so that we can recombine them to produce a recombinant DNA, the most important element in the gene cloning is the uh, carrier molecule called as vector, which in conjunction with the insert DNA or a foreign DNA or a passenger DNA, it recombines to form a recombinant DNA that can be propagated into a host cell. So here you can typically see the whole process of recombinant DNA technology. The basic steps being isolating the target DNA from the source organism by using a PCR or specifically cutting with a restriction enzyme. And you need a carrier molecule, a vector for carrying your foreign DNA into that. So these cut DNA from the source as well as from the carrier vector can be joined together by using a ligase enzyme that will produce a recombinant DNA. This will be mobilized into a bacterial host system or a yeast or any other biological host system for propagation. So every time when this recombinant DNA multiplies and produce a recombinant protein, its own protein, it also produces the recombinant protein, where you can typically see the expression of the recombinant protein in the cloned host gene. So this recombinant DNA or genetic engineering has umpty number of applications. They have basic applications in nucleic acid manipulation, where you can do antisense RNA derivations or antisense RNA technology to suppress the gene expression. It is also used in gene therapy and producing vaccines and also in molecular diagnostics. In case of microbiology, we specifically use this recombinant DNA technology to produce recombinant proteins using the microbes as a biological host or factories to produce vaccines or peptides, small molecules, therapeutic proteins, industrial enzymes, and biopolymers like plastics, bioplastics can be produced using microbial host systems. And similarly, this technology has applications in agriculture to produce transgenic plants expressing foreign protein, which aids in resistant against insect pest and pathogens, as well as for improved quality, as well as improving the shelf life of fruits and vegetables. The plants can be accordingly manipulated by using the recombinant DNA technology. In animal sciences, this technology or gene cloning recombinant DNA technology is used to produce transgenic animals, which can aid in producing therapeutic proteins, or you can also go for strain improvement or improving the breed of the animals. And this also can serve as a disease model for various other applications. So together, we have discussed on the DNA manipulative enzymes, in particular, as one of the tool for recombinant DNA technology. Thank you.